Ladies and gentlemen, welcome to the Quarter 3 elective undergraduate course, Selected Topics in Biopsychology. I'm so glad that I have this quarter and I will have this chance to lecture on this topic. Some of you already know me, some of you do not know. For those who do not know, I am a molecular neurobiologist by training and also neuroscientist, which gave me the opportunity to really study behavior from a biological perspective, but that means you have to work interdisciplinary. When I explain you biology of behavior, you see that you cannot just study how you study cancer cells or how you study bacteria. So you have to deal with philosophy when necessary, or you have to deal with a lot subjects actually, and that will be also reflected hopefully with this lecture today. So human brain are made up of billions of cells. They did some kind of, they used some methodology of cell counting, and they ended up like an approximation like 100 billion cells. And these cells are interconnected with each other by synapses. Synapses are subcellular structures that mediate nerve signals between neurons. These signals are electrochemical signals by nature. And the question is, how such electrochemical signals that are mediated between the neurons, how the activity and interaction of all these neurons are giving rise to the behavior. And not only behavior, the behavior itself is a very huge topic, but think about the behavioral diversity of human beings during health and disease. So that's a tremendous topic. So that is the topic of our course, but this topic is not an easy topic, and this topic yet not answered. And in fact, how does it happen is one of the most challenging questions of our century. So be ready to be curious and challenged all the time. So, oops. I think I pressed to the wrong place, or yes. So biological psychology or biopsychology, behavioral neuroscience, or behavioral biology, they can be used in the same meaning. In this course, I will mostly use the term biopsychology. So now, what is biopsychology? So the study of biology is the study of psychology based on biology, but it's an integrative discipline, not only biology. But as you see here, biology itself, where is the cursor? Yes. Sorry. As you see here, biology itself has so many different subfields. That is also not just something ordinary that happened in the last 50, 60 years, that biology has been differentiated into so many disciplines, and those subdisciplines have already become something by itself. So if you ask me, I am a molecular biologist, but there will be somebody who works with the plants and who is maybe working with plant physiology, and he could also work with plant molecular biology. Or somebody who is working in the cancer, he is actually working in the field of molecular biology, but in cancer cells. But the molecular biology is molecular biology, and I use the rules of molecular biology and other associated techniques for neurons, the nerve cells that are found in the brain. But of course, you cannot think molecular biology independent from genetics. Oops, sorry. Anatomy, physiology, and behavior. Here, especially anatomy has a very specific importance, because not only in the molecules, you will see maybe if you are curious in the future and take some biology courses, you will know that, for example, the structure of proteins are very important for the function in the cell. Just like that, the structure of the nervous system has an important reason for its actually application and function in the nervous system. So anatomy itself is very, very important topic. And of course, there are lots of other fields that are associated with biological psychology. I will not go through with them about that. I will not go one by one now for today, but later we will have more chance to discuss. So, what is the style of biopsychology? What kind of scientific approach does it have? So, as I already said, it's an integrative discipline. As I already, no, I didn't say, now I'm saying, it's a pure science, but both, it's also it's an applied science. Meaning that, you know, pure science, some people who took the course from Problems of Modern Science, from me, they already know what is pure science. Who would like to answer? Pure science?
I mean, just theoretical science.
Pure science?
Or like quantum mechanics or...
I wouldn't say theoretical. I think our students shy to respond, but the pure science is a science which has a main motivation, just motivation of intellectual curiosity. So the researcher is curious because researcher is intellectually curious. Why does it happen? There is no any utility, a reason for a utility for that. On the other hand, applied science are focused on solving practical problems for human beings. For example, all engineering fields are applied science. Biology is a pure science. Biotechnology is an applied science. Medicine is an applied science. And biological, on the other hand, biopsychology is using both pure both pure science and applied science. And in biopsychology, we also use both top-down and bottom-up approach. What is that? In the top-down approach, think about patients of depression and think about normal people. You separate them and then you take some blood samples and you compare, which has been done so many times, and eventually it was understood that it was not the best way to understand the biology of depression. In the bottom-up, but we still use it in a modified way. In the bottom-up approach, on the other hand, for example, and of course these approaches are working with interaction with each other. So, for example, some findings that have emerged from the top-down research can be further studied by bottom-up approach. For example, you identified a gene variation that is more common among the people who are depressed. Then you want to find the equivalent of that gene in a mouse, and then you can just delete that gene in the mouse genome, and that is called knockout mouse. And then you study the function of that gene in the phenotype of the organism. So you start from the bottom, from the gene, and then you come to the behavior to analyze the outcome. Also, and these were so far experiments, but biopsychology also using case studies that has a very special context in the biopsychological research, and I think I should really mention that, the case of HM. So, you know, during the medical conditions, when people go to the medical doctors, sometimes there are very unique cases of patients, and then they study that patient's case in a collaborative way within a team. And that was perhaps the most famous case study in the history of neuroscience is the patient HM. So this guy, at that time in the 1950s, 27-year-old, and he was suffering from seizures. And eventually, his seizures became so bad, he was suffering so much, that he decided to undergo a surgery. So what happened? That as a result of the surgery, they have, this is a normal brain, this is the normal brain, and they removed the medial temporal lobe, these regions, in the brain of the patient as a result of the surgery, and hoping that his seizures are going to be gone. And after the surgery, yes, his seizures were dramatically gone, and his IQ was okay. He was totally fine. That's also very interesting, like, you know, some part of your brain are removed and you're okay, and seizures are gone. But something very dramatic happened to him, that he was not able to form new memories. Interestingly, he was remembering everything before the surgery. He was only not able to make new memories. So this experiment, and this is famous Brenda Milner. Dr. Milner was working with this patient for 30 years, and it was her studies lead to the identification of the fact that there are different memory systems, and hippocampus is involved, because in this medial temporal lobe there is a brain structure called hippocampus, and it was one of the reasons why we know that we know the fact that hippocampus is involved in memory and learning. But when he was meeting with Brenda Milner every day, not every day, maybe a few times in a week probably, but for 30 years he was not able to remember her. He was not able to remember her name. Each time she had to introduce herself to him. Actually, there must be a whole lecture, even a few lectures, to explain which kind of studies have been conducted on HM. That is just for me today is an example, but believe me, as a result of the works done on HM, today we also know that there is unconscious learning, an idea that was already proposed by Freud long ago, that there is this, we have this conscious state, but also we have something subconscious, like, you know, when Freud says subconscious, as if like there is a physical state which is a little up and down. But actually, in science, we don't speak that way if we don't prove it scientifically. But still, I mean, what is up, what is down, according to what? But just to remember that, still, I think the idea of Freud to think of a subconscious state and unconscious learning was proven by some of the studies of Brenda Milner. So that's why case studies are very important for actually biopsychological research. Biopsychology is a subdivision of neuroscience, and the neuroscience is scientific study of nervous system. So that's why sometimes I can use the term neuroscience, because it is related to biopsychology. So in this slide, I would like to explain the impact of neuroscience on psychology. But I want to maybe give you a very direct impact, maybe the ones that students can perceive in a better way. And that is this slide. So psychology departments change their names, and psychology programs start to offer Bachelor of Science degree in addition to Bachelor of Art degrees. As you know, what is Bachelor of Art and Bachelor of Science? So normally the social sciences give you a Bachelor of Art degree. But now, especially in the American universities, you observe a trend. And that trend is now psychology departments change their names. They don't call themselves as psychology anymore. They wanted to say, for example, Department of Psychological and Brain Sciences. Or they want to also add Bachelor of Science degree, in which is also a psychology major, but in a Bachelor of Science degree of psychology, you have to take a lot of biology and statistics and maths. Why there is such a shift? Why there is such an identity shift? Because of the developments in neuroscience. Okay, as a neurobiologist, I really adore this, and I feel like responsible to really express this spectacular work, which was done more than 100 years ago by Santiago Ramon Cajal, the famous neuroscientist. So at that time, with very limited opportunities, with a very basic light microscope, but with the use of a technique called Golgi Staining, he was able to actually visualize the cells of the nervous system from different parts of the brain and draw them with his pencil or pen on his notebook. This is one of his drawings that was done in the 19th century. And here, this is the retina shown in this figure. You know what is retina? Retina is involved in the vision. So, but it is actually anatomically belonging to the brain. So sometimes they say when you look at the eyes of the people, you look at their brain. So retina has many special cells, and one of them is actually photoreceptors. Those photoreceptors have the ability to take the light reflected from our environment now. For example, you look at me and you see me, you see your surrounding, and all this thing happens because the light reflected from the objects around you just come to your retina, and there these cells, these photoreceptors, convert this light into a nerve signal. So they actually make a kind of signal transformation. They transform a light signal into an electrical nerve signal. Then, in the whole retina, you see the cells that are beautifully described by Cajal's drawing, and you see that group of cells that are connected vertically with each other, and then there are actually horizontally aligned cells which are also making fine connections with each other. And from there, these electrical signals are transmitted to the brain, to the visual cortex via the thalamus in your brain. And by that, you are able to see. Another beautiful drawing of Ramon Cajal is from a brain structure called cerebellum. We will learn about it, but cerebellum is found here in the posterior part of your brain. Here you see he's not only a scientist, but he's an artist. And you see one of his beautiful drawing of beautiful two kinds of cells. This is, for example, how big is it? This is the cell body, and these are all very complex processes called dendrites. And then these cells are actually one of the biggest cells in the nervous system called Purkinje cells. And then you also see granule cells that have long fibers connecting with the dendrites of the Purkinje cells. So these are all shown by, drawn, studied by Cajal, and that was very, very significant at that time. So why do you think I was talking about Cajal's anatomical drawings when I want to talk about behavior? Cajal at that time, by studying those cells, he actually made a very significant proposition, and that was, he was thinking and proposing that perhaps the memory is actually encoded by the structural changes between the connections between the neurons. Look at this idea. So the memory, learning something so abstract, occurs as a result of structural change in the brain structure, in the neural level. So Cajal was proposing this idea more than 100 years ago. And that was a great opportunity for psychology, right? Maybe to look for a behavior in a physical substrate. But let's have a look at now other dimensions. Now you see here especially two famous scholars. I think everybody realizes Mr. Freud, and on the right, and on the left, Emil Kraepelin. So these guys were studying madness. So madness actually was not the subject of scientific study until late 19th century, when these guys decided to study on them scientifically. And for them, for Emil Kraepelin, madness was biological. On the other hand, for Freud, it was psychological. Although Freud, in some of his books, mentioned that some sorts of mental illness could arise from biological processes, still, he mostly formulated those disorders as the disorders related to childhood experiences and psychic forces. On the other hand, Kraepelin was relying on a very strict biological reason behind of the mental diseases. And these guys were living in the same century, both speaking German, but somehow they never met. And their also ideas somehow go in parallel. They never cross each other for some reasons. But now I would like to take you to the 1950s, after the Second World War. What was the situation in medicine? I think this paper is a very important paper. This is a paper published in November 25th, 1949, just 1 year after my father was born. So, and this paper very well explains that at that time, this is an indication that medicine is being transformed into a molecular, biologically based field. So, sickle cell anemia, a molecular disease, has been described in this way. And let's have a look, and medicine now is already amplified this start with the progress since then. I don't want to go too much on that. We are now in the age of personalized medicine, in which you actually want to diagnose and prescribe the patients based on their personal genetics. What about the situation of psychiatry in the 1950s and 1960s? I would like to just read this sentence from Eric Kandel, who is himself a psychiatrist and also actually a psychoanalyst, and also Nobel Prize winner in the field of medicine, because he found the molecular biological basis of learning and memory in animal models. So what does he say? In the 1950s and 60s, academic psychiatry somehow abandoned its roots in biology and experimental medicine and evolved into a psychoanalytically based and socially oriented discipline that was surprisingly unconcerned with the brain as an organ of mental activity. So that was something very profound, because that caused also somehow psychology and all associated fields to be perceived as a social science. But now I would like to give a little break into this debate, and not debate anymore, but the history. Now I would like to talk about a little behavior. What is behavior? So according to the dictionary, it is called the way in which someone conducts oneself. Here you see a very different patterns of human behavior. I mean, look at that child especially. I mean, children have their own nature, that is depending on your developmental stage. You behave sometimes differently as you grow up. That is interesting. Look at these guys here. There are so many different components of this behavioral phenomenon here. Here there is a group work. That means it's a teamwork. I mean, it looks like it depends on a lot of cognitive skills. So they are thinking, probably using the prefrontal cortex. And there are a lot of social skills going on. There is a hierarchy maybe. For example, this guy seems to be more senior and explaining what to do next, and they cooperate together in a team. So very complex patterns of behaviors are taking place at the same time. And what about this? So, or what about that? The crime. So, but I would like to also show you something more fundamental. So, when we think of the behavior, maybe let's take something very common, no matter who you are and where you are and at which age. This is your main thing, and that is you drink, you eat, you reproduce, you sleep, and you excrete your metabolic waste, and that is a cycle. Actually, you wake up, you survive, and you sleep. That is our life. Now I would like to take your attention into this toilet paper, which is pink. Why do you think it is pink? What does it mean? Any idea, anything?
It's going to be white, right?
Yes, but I chose pink by purpose. I think white represents, even the colors have something for human mental perception. So white represents cleanliness. That's why we want it white. I think nobody would like to have a black toilet paper. I don't know. Maybe some people as a variation. But also pink still looks white, clean. It gives like, you know, soft, innocent, you know. I don't know. It's like, I don't want to give so many meanings to toilet papers, but some people obviously were thinking about that, thinking about the meaning of life. Who is this guy? So I want to ask the Generation Z in the classroom, the young generation. Yes. So for him, of course, the life was absurd, huh? The whole thing. And this is like, if you eventually, you can reduce everything into this cycle. And this cycle actually is not so simple or unimportant. For biological reasons, that is a cycle required for your homeostasis. Homeostasis is a tendency for the body to keep an internal environment that is required for optimal survival. And all this cycle is, and everything is working for that, for our survival. I would like to really mention this pink toilet paper detail into this novel when I read in my youth, because he was mentioning this life and how we actually start as a human civilization, starting from the beginning to the end. He was saying, so I think he was actually relying on the deterministic philosophy and then combining it with this anxiety of life. And he was in this novel, maybe you guys don't know, but in my youth we know that. So he was in one of the part in the novel, he was explaining that this pink toilet paper is not something you should ignore. It is about the meaning of our life. That we try to, you know, forget this anxiety because of all these details. But at the end, that is the absurd life. Of course, some people don't think about that at all, but obviously some people think about that, and it has such an impact that, for example, there was a field called Existentialist Psychotherapy. So in our class, we will not talk about this, but we will definitely talk about biology or molecular biology of mental disorders. But I would like to say that how complex that phenomenon is that. It's not like as simple as understanding or diagnosing, maybe. I wouldn't say understanding, but like a diagnosis of cancer. Now I also would like to mention mind, because when we think of behavior, we tend to think of things that are maybe more visible. But if I say mind, and then there is consciousness, perception, memory, imagination, language, intelligence, emotions, judgment, all these different forms and faculties that require cognitive or non-cognitive activities. And they are all stemming from the brain. Now, let's come to now, 21st century. What is our state? After all these long journey of the brain sciences, or understanding of human behavior and pathology of the brain. Now, what is our situation? Luckily, we are in a good time for understanding of brain and behavior. For example, now brain-machine interfaces have become possible. Now there are a few cases already in the literature, in the medical practice, that the totally paralyzed patient from here until the legs can walk by just thinking with a machine that is actually integrated with his brain. So, like thinking and moving with the help of a specific exoskeleton that is now possible in our century. And the techniques in molecular biology, such as optogenetics. Have you ever heard of optogenetics? It's a technique by which you actually insert some genes that have the capability to produce proteins that are light sensitive. And then you insert, you target light to those proteins that are found in cells that you put by yourself, and you modulate the behavior of those cells by light. And if those cells are found in the live organisms, then you control the behavior and physiology of the organism by light. And that is already being done. So, together with the Fourth Industrial Revolution, you know, this is a slide from one of my previous courses. For those who take these problems of modern science should know this slide, right? They remember this slide. So what we have genetic engineering, we have Internet of Things, 3D printing, virtual reality applications, quantum computing, and believe me, every bit of these developments have an application in the brain sciences as well. And that's why we have now incredible opportunities to study the brain. So that's why I would like to say that, yes, it is a challenging question to understand the biology of behavior, but we are in a good time. We are in a good time, in a good place to deal with that problem. So now we start to the second part of our class, and that is the biological model of behavior. What is the biological model of behavior? Anybody has any opinion? What kind of components would you expect to be? Yes? Yes.
I think maybe that's about the reaction that we go with.
About what?
Some chemical reactions inside our body that affect our behavior. Like it's
yes.
Not the external factors, but something from inside.
Okay, you said not external factors, but yeah, that's a very important point, because in this class we will learn that actually we don't really separate external factors and something coming from the organism. We are looking for their interactions, because both of them are required. I think in the previous class one of you was asking that. That is the nature-nurture debate, but it's not a debate anymore. Nature and nurture, genes and environment interact, and they are all required. So in this figure, you see the current model of the behavior for biological perspective, according to biological perspective. So the genes we have are selected by evolution, and it is those genes that sculpt the brain structure. And there is a reason for that. They are actually serving for our survival and reproduction. And the experience is extremely important, but when I talk about experience, it's not only, I don't know, how you were raised. It is everything everything since you are a zygote in your mother's uterus. So it is the food that you ate, diet, all the chemicals you were exposed, diseases you had. It doesn't matter, it's a biological agent. Something coming from outside is an experience, and it could be biological, social, economical, psychological, everything. Okay? And experience is required for the development. There are certain periods on for human development that it is required for brain. It's a guide. So certain, for example, vision, light signals have to come to the, after the birth, it has to come to the eyes of the infants for suitable development of the neural connections. Otherwise, it cannot connect. It needs guide. You know, when the brain is formed, the connections are done, but they need to find refinement, a refinement. And that refinement requires a guide, and that is environment. So we need environment. And that shapes the brain, that adjusts in the finer way the brain connections. And that is also a process called neuroplasticity. Neuroplasticity is a process by which the brain changes as a result of experience. And that neuroplasticity is much more high during development. That's why everything what we have during development is more important. So, as a result, the current organism is a total outcome of the genetic and experience or environment-dependent factors. So you perceive your current situation according to your current brain. So how is the state of your current brain, which is the result of your genetic and experience-dependent situations. So then you respond according to that. And there is a feedback also in this paradigm. So we will have more chance to talk about this in more detail, but now I would like to speak about this bit a little, but not too much. Just I would like to give a little history. I am sure all of you are already familiar from the secondary school that, you know, principle of inheritance was discovered by Mendel's experiments. But those principles, although they were done in plants, they were actually, you know, they were actually instrumental and fundamental for the rest of the organisms. Although there is a Mendelian genetics, which is discovered by Mendel, but there is also non-Mendelian inheritance patterns as well. But that part, at least that is a part that was discovered by Mendel, is actually applicable for the rest of the living organisms. That is not surprising. Why? Because, you know, we have all these different organisms with all different genes, but one thing is common, and that is the central dogma. What is central dogma? In high school, I think they are teaching central dogma as protein synthesis, right? We call it sometimes central dogma because it's universal. It doesn't matter which kind of living thing you are. So it happens in the onion cells, it happens in brain cells, cancer cells, bacterial cells. And that process is that there is this DNA, and DNA have actually gene regions that are responsible for the production of specific proteins. So this is the, you know, DNA is transcribed into messenger RNA, and the messenger RNA is translated into a polypeptide, which is the primary protein structure. Then the protein folds and gets into its final 3D structure and does all the functions that is required for the cell. So there are so many different genes. That means that as many as proteins, even more. So let's have a look at the humans. Humans have about 22,000 genes, and they have at least this amount of proteins, but actually more than that, because sometimes genes encode more than one proteins as a result of some mechanisms called alternative splicing in the messenger RNA level, for example. So here, what I want you to understand when I speak about the behavioral phenomenon, when I talk about the gene and what does a gene do, you have to think always in terms of the product of that gene, and that product is the protein. But that protein is encoded by genetic factors. That's why we always talk about the genes, because they produce the proteins, but the proteins do the cell function, and then they generate a physiological state. Yes?
Why is grape so special?
As you see with this figure, there is no actually correlation with the complexity of the organism and the number of genes. That's why they put it grape by purpose, to show that that is not correlated according to, our complexity. So, but I also would like to show this now, because now, today, we are living scientifically in the post-genomic period. What is post-genomic period? So anything that is after the release of the Human Genome Project. So what is Human Genome Project? The Human Genome Project is the project by which every little nucleotide that is making up the DNA of human genome has been identified, sequenced, ordered, catalogued, and uploaded into a database which is open publicly. If you wish, we can explore that later together in another session. And that is another opportunity for biopsychology. Now I would like to speak one important topic, and after that, because that is necessary for us to understand our specific subject, crime. Because I would like to discuss with you what is the reason that why some people are involved or why some people commit crime. For that, you need to know this factor, and that is genetic variation. So what is a genetic variation? We are not same genetically. We have a human genome, but human genome have different versions in each human. Each person have a unique inheritance. And that unique state is coming from the what? From the genetic variation. So we inherit one version of a genetic variation, and that variation is amazing. And one source of that variation, there are others, but one of them is called single nucleotide polymorphism. So what is that? If you can see here, this is the person 1, this is the person 2, and this is a sequence, genomic location of the same genomic location of 2 people, and it is actually encoding for the same gene, but genes have variations. And that is because, for example, if you look at, there is a cytosine here and there is a thymine here, and that's a variation. And that variation also, all these kind of variations have already been also identified in different human populations, and they are also catalogued and uploaded publicly available databases. And some of these variations are inherited as groups to the next generations, and these are called haplotypes. They are important for understanding the genetic background of diseases, and that was depicted by a project called HapMap project, and that is also now publicly available. If you want, you can search it. Another important genetic variation is, for example, variable number of tandem repeats. Don't bother yourself with this term, but basically just look at this pattern. So these are different. This is one unit of a genetic sequence, and 1, 2, 3, 4, 5, 6 of it is in a stack in one person's DNA. In the other person, 1, 2, 3, 4. In the other person, it's in a different location, and 3. In another person, so this is another form of variation that is naturally found in our genome. So these variations are naturally found. Okay, for any gene that you have, in any those 20,000 genes, you have different variations that are reflected by these kind of variations. So why did I mention that? Because I want to show you one important thing, that the majority of the behavior we have is due to the complex genetic and environmental factors and their interaction. So that means that if you want to understand the behavior, you have to understand the brain and gene-environment interactions on it. And some very important studies have been done in this field, and that's why I would like to discuss one or two of them according to time. I don't know what time is it now.
8:10.
8:10. We have time, yes. Okay, I'm glad. So you know this guy, right? Who is he? It's written. I mean, I sometimes really like to share movies and novels in, you know, for our students, because we have very different groups of students which I really like, from, you know, cultural studies, from students who study literature maybe, or visual arts. So this movie and this character here, Hannibal, okay, so I don't know if he's described as a psychopath or not. Do you know? Is he a psychopath? Or what is a psychopath? Have you ever heard? I mean, in popular terms, you are not asked as an expert, but when I say a psychopath, what do you ever call somebody psychopath when you get angry with them?
I mean, if I'm not wrong, a psychopath is the one who commits crimes based on a specific, you know, way, or like has a specific motive to repeat the same crime over and over on different people. Right?
So they do crime, you think?
Yeah.
No, I'm asking your opinion. There is no correct or incorrect, because I want to know the popular meaning, not the expert meaning of a psychopath. So somebody who commits mostly crime, you think is a psychopath?
No, not just a crime, but he has, you know, a specific reason for that. And
Reason?
Yes, and based on this reason, he would commit the same crime.
Same crime, like a serial killer.
Oh, maybe I'm just making it up and confusing it with a serial killer, actually.
You wanted to say serial killers?
Yes.
Okay. Any idea about the psychopaths?
No, I have a feeling that it's probably just a colorful understanding of that. Right? The psychopath is someone who is outwardly normal. I mean, he's a classical psychopath in Hannibal lecture. It's not that he's depressed or he's neurotic. I mean, he's a normal person, but he has something in his brain that just makes him eat people, right? Otherwise, he looks normal, but there is one trait that he cannot get rid of. It's part of his nature that makes him behave in a very unusual way in certain situations.
Yes, and yes, you wanted to say?
If I'm not mistaken, psychopaths have antisocial and narcissistic tendencies in their behavior.
Yeah, these are all the terms. Of course, they are used in medical practice for diagnosis, and that is good for them. But in the popular terms, we use it for everything. For example, when you feel low, you say you are depressed, but maybe you are not really depressed. I mean, narcissistic personality disorder is a case not necessarily to be associated with always with the psychopathy, but let's have a look at the definition, official definition. According to, you know, Diagnostic and Statistical Manual of Mental Disorders, this is the general guideline textbook that is used by psychiatrists, okay? So that is our guide, and according to that, disregard for and violation of other rights since age 15. For example, age is important. So obviously you don't become psychopath. If you have not become psychopath until now, so you're okay. So by one of the 7 sub-features, failure to obey laws and norms, by engaging in behavior which results in criminal arrest or would warrant criminal arrest. So that is very, you know, just one of them. But I just wanted to read just one, you know. So there is a strong correlation between committing a crime and psychopathy, but that doesn't mean that whoever is in prison now is a psychopath. We don't say that, of course. So, but, you know, for researchers, we are more interested in what makes somebody a psychopath. Okay, so people study their lives, why these people become a psychopath. And they found one common thing, that the children, especially boys, that is important. When I say boys or girls, when I separate and associate a certain thing, that means that there is a biological relation, because gender is, or sex, biological sex is determined by your genes. So this was a pattern that was especially observed in boys, okay? So boys who experience familial adversity such as physical abuse, interparental violence, develop behavioral and emotional difficulties, and one of them is actually the risk of developing this kind of antisocial behavior. But interesting thing is, there were also cases, like there were also some boys that were not actually developing this pathology. So no matter what kind of abuse, whatever they had, they don't turn into a monster. They don't become a serial killer. They don't become disrespectful to other rights. So why? Why somebody with such an abuse turn into a psychopath, while the other, despite all negative factors, do not turn into a psychopath? What is the reason? So I would like to show you actually a very important study that was done. They followed the lifestyle of children starting from age 2 until the adulthood, who were also abused. And that was published, I think, in the beginning of 2000s. And that was also replicated by some other studies. That's why it is a very significant study today to discuss the crime and gene-environment interactions. But before that, first I would like to introduce you some basics so that you can combine the puzzle. So the gene in our topic today that we are going to discuss is the monoamine oxidase A, the gene encoding for a protein that is called monoamine oxidase A, meaning that this is an enzyme, and the gene encoding for this enzyme is located in the X chromosome in the human genome. Okay, this is the genomic location of this gene. And what does this gene do? As I already said, this gene is encoding for a protein called monoamine oxidase A. It's an enzyme, and what does it do? It is involved in the breakdown of certain brain hormones, which are called neurotransmitters. These are dopamine, serotonin, for example. And there are naturally two versions of this gene in our populations. So one version leads to the production of a low activity monoamine oxidase enzyme, while the other version leads to the production of the high activity monoamine oxidase A enzyme. And consequently, the brain level of these neurotransmitters, such as serotonin and dopamine, will be modulated according to the gene that you have. Okay? That is a naturally transmitted case, and that doesn't mean that you are sick if you have one version or other version. I am explaining a normally, naturally occurring diversity of a gene and its consequence. So they wanted to study the monoamine oxidase A gene and its association, the variation of this gene and its association with antisocial behavior together with the childhood abuse. This is the description of the monoamine oxidase enzyme, and these are the neurotransmitters that are actually metabolized by monoamine oxidase A. already in the previous studies, they have found that, for example, if you have high monoamine oxidase A activity, okay, the monoamine neurotransmitters will be lower, and in the humans it will have, it may cause anxiety disorder. Not it will, okay, it may. And in the animals, they wanted to see what's happening in the animals, and then, you know, they find the gene encoding, because animals have this gene too, mice have this gene too. So they delete that gene in the mouse genome, and they develop a knockout mouse, knockout animal. A knockout animal is, as you can understand with the name, is an animal that does not have a particular gene on the organism, but it's live. And then you study the behavior, and you observe anxiety-like behavior compared to the control animals. When you have low activity of this enzyme, then those neurotransmitters are quite high in the brain. And that causes excessive aggressive behavior, and also in the animals, some other problems. So, according to the study that I will introduce you in a few slides, so they took more than 1,000 male subjects, okay, from childhood to adulthood. This study was done, I think, in the United Kingdom. And then they recognized these children, especially because they were abused between the ages from 3 to 11 for some reasons. And then they screen their lives for, you know, for the 30 years. And they look for an aggressive behavior by the age of 26. And this is the result of the study, and this is the study actually, Caspi and his colleagues, which was done in 2002. Here in this graph, what you see, here, in the vertical line, you see this is a measure of the antisocial behavior, and in the horizontal line, you see the degree of the childhood maltreatment. So there is none, probable, and severe. And the relation here, there are two different lines. The gray line shows high monoamine oxidase activity, and the black line shows low monoamine oxidase activity. And you look at just the sphere, take a top from here, which is the highest score of psychopathy, and then go down. You see the difference between these two. So when somebody have low monoamine oxidase activity, which is due to the gene having that variation, they are more likely to develop antisocial behavior if they are exposed to severe childhood abuse. So that's a very good example of gene-environment interaction. So what we conclude from this study, we understand that we have genetic variations, and some of these genetic variations can make our brain vulnerable to certain environmental adverse effects, at least for this case. And if you are also environmentally challenged or exposed to such a situation, then you may at risk of developing a, for example, state like this. Okay? So that is the reason that we actually really focus on the gene-environment interactions. We don't separate it from each other. There are many other experiments, but this is a rather new experiment. And also this is replicated by meta-analysis and some other studies. That's why I really adore this study. I really like that. Any questions so far? Yes?
Does that mean that we should put a person with severe maltreatment and low MA or R in prison just in advance?
That's a very good question. That's a brilliant question. Actually, that is one of the things that I would like to discuss with the students today. But before that, so that is, I think I already explained that, but let's come to your question. The crime genes, for example, can we say that now can defense lawyers can use the genetic background as an excuse to rescue the people? That happened actually. I would like to show this guy. This is the first case. It happened in the United States in 1990s that Stephen Mobley, okay, he committed he killed, I think, when he was visiting a pizza shop one day. So at the age of 25, he walked into a pizza store and casually shot the manager in the neck after robbing the till and joking that he would apply for the job vacancy when the man was dead. So here is a list. The first one is the Mobley case. So when charged with the murder during the trial, his lawyer asked for genotyping. They say that, oh, you know, you don't know about these studies. He is actually carrying a gene which makes him vulnerable to this, committing a crime. However, it was refused by the court. So he was executed. But the second case was happening, and then he was actually asked to be genotyped, and then court was accepting that, and he was genotyped, and he really had the risky version of the gene of monoamine oxidase A. Plus, of course, that is not enough, but he also had a childhood abuse, according to recordings. So he has everything, according to the previous paper that I discussed. So what was the result of the court? He still took 32-year sentence. I don't know if he would normally take even more sentence. I don't know. But the third case, this was in Italy, I think. This was an Italian case. In that case, sentence reduced 1 year because of the genetic evidence, although actually the adverse environment was not clear. So you see, actually, the judges have certain very different attitudes. So the question you ask Andre is still under a debate. So even with the behavior of the judge. But
my question was, I mean, logically it would make sense to isolate these people before they committed the crime.
Yes, I would suggest this. Actually, it is at the end. So we usually tend to, of course, to show the case examples I wanted to show you because I want you to see how these developments are introduced into the legal practice. But in the scientific arena or in the from the medical standpoint, maybe we would like to see this from this point of view. So if we can understand the risk factors associated with genes and environmental factors, and once we are aware of these individuals who are vulnerable, perhaps we can do some protective, some social and medical protective mechanisms can be evolved in order to protect the generation of antisocial behavior. However, I think we still need a lot of scientific evidence to really develop a model for that. But I think we are going into that direction. So that is also one indication of protective medicine. You know, medicine now is evolving from a practice, evolving to a practice that is personal, predictive, protective. So these can be used to protect the diseases before they occur. And that is a good example for that. But I think it is still very early for this kind of application in reality. But I want to show you this. This monoamine oxidase A gene has become so popular and so popular that today in the market you can order a home use test. So that is a test for normal consumers, not the specialists. And you can actually do and test which version of the monoamine oxidase A gene you have. So that is already in the market available. And they call it, in a, for some people, it's an advantage. If you don't have a childhood abuse, and if you have this gene, you are a little more aggressive, and maybe it is good if you are a professional football player or a soldier. Depends, I don't know. So maybe I don't know each of us which version of that we have. That's an interesting question, right? But here, as I said, it doesn't matter even if you have the risky version of this gene, because in order to develop a psychopathology, you need to have an adverse environmental condition altogether. So genes do not make you ill. For psychopathologies, they do not make you ill. Maybe this risky gene for the antisocial behavior could be advantage for another kind of behavior. You never know. So you have all different repertoires inherited, and they do not make you sick, but they can make you vulnerable to certain diseases in the case of bad environmental factors. And for each person, it could be different. So in this case, we study antisocial behavior. But now, I want to discuss with you something maybe a little sweet, cute. With the laboratory animals, I by myself work with mice in the lab. You know, the mice is the small ones, and rats are the big, very big ones, but they are white and they look like a rabbit and very cute. But no, the mice are very small. They are usually black. To me, always they were more cuter, actually. And I want to show you this, just for those who are not familiar with this kind of research, that is also something in the biopsychology. Okay? And that is a cage. And there are different kind of maze. Maze. And there are different kind of mazes. And you test different kind of abilities of the animal in these mazes. And for example, learning and memory abilities. And usually, you know, you see the animal has to navigate. Once you leave the animal there, you record it, and then you really have the whole apparatus to measure the time and length that the animal spends in the maze while the animal navigates the maze. And usually there is here a reward, which is a food. And you know, then of course they want to find the reward. I mean, they are not aware maybe, but by chance they find it. And then next time you put it again, and next time you put there again. And how long does it take for the animal to remember the location of the food in the maze? So do you think some animals can learn quicker than the others? Some mice can learn quicker than the others the location of the maze? Some people were curious about that, and they did an experiment. And that is a very famous experiment, which is a textbook example. So that is done in the 1930s, study of Tryon. So Tryon working with the maze and the mice together, and realized that some are just more talented in one task. That's very important. So when I say here, so that slide is about actually genetics and environment's effect on the intelligence. But intelligence is very complex, and here we just take one test, and just it cannot be considered something like a whole. Just one thing that is in the maze, and you know, for other things maybe it could be different. That's very important to understand. Intelligence is very complex. So it depends on the criteria of your test, what is intelligent. But for this case, let's say, because we have to find a way to test the animal's ability to navigate the maze, and you know, not we, of course, I'm in Tryon in the 1930s. So this is the original population of the mice. Here you see the percentage of population, and this is the percentage of population, and the horizontal line shows you the number of errors. So when you put the mixture of different mice, let's say 50 mice, I don't remember the exact number, and then you measure the number of errors for the animal to find the reward in the maze, and you record them one by one. You see, for example, in the animal population, there are different animals with different kind of errors. They are not the same. They are not showing the same performance. So Tryon separated the animals according to their performance, and they divided them into two groups, maze bright rats and maze dull rats. So this is the group of the mice that are very good in that show a better activity in the maze test, did less errors to find the reward. And this is the animals that are not so good, that they were not so well. And then they are separated, and then selective breeding is one of the actually methodology to study genetics. So these animals were separated from these animals, and these are the maze bright rats. They were crossed only with each other, while the other ones were crossed only with each other. So they were not allowed to mix and reproduce, but they were only reproduced by themselves within a group. And after 8th generation, they have actually tested their performance in the maze, and they have found that the offsprings of the maze bright rats are really very good and doing much less errors compared to the offsprings of the maze dull rats. So this study shows the effect of inheritance on the intelligence, at least for this case and for this animal group. But what is interesting here, because so far we were only discussing the genes, but I promised you to talk about the gene-environment interaction, so I would like to mention you another experiment, which is actually this one. This study was done by another group in the 1950s, and they actually wanted to test the effect of environment on maze bright and maze dull rats. Think about this. You have the maze dull rats, and what if you give them an enriched environment during development? Do they become smarter? And what about the maze bright rats? Are they going to become super smart? What is your hypothesis? Or nothing will happen? What do you think? Yes. What do you think?
Well, the scarce food supply or whatever.
So good food
will make them work there.
So you think the smart ones will become even super smart?
Yeah, maybe.
Maybe. Okay, let's see. They did such a study, and they were also wondering what will happen. So this is cage. A cage is a standard cage, is described like this. This is the cage we use in the laboratory. So the animal is kept in such a standard environment. And this is an example of an enriched cage. That is very important. Also, you have to think this in terms of children as well. During development, children also need stimulation in all way, in a positive way. And this is the same kind of thing for these animals. Look at all these different kind of toys and colors that animal can navigate and play with during development. Okay, so they divided the animals. They took the maze bright rats and maze dull rats, and they are actually grown in the enriched cage like this. And also maze dull rats and maze bright rats were also raised in the impoverished standard cage. And they were actually wondering the results, what will happen. And that is what they found. So this is very interesting. Oh, this is a wrong one. I am sorry. I will explain you verbally the results, and later I can show you the graph. What they found very interesting, they found that in the enriched environment, maze dull rats become as good as maze bright rats. However, maze bright rats do not become better. They do not become super smart. They are as good as before. They were maze bright, and they are still maze bright. But maze dull rats, with the enrichment, with the environment, they become as good as the maze bright rats. That was the result of the study, which shows the effect of environment. So you can conclude a lot of things. For example, you can say that, oh, why the maize bright rats, despite the enriched environment, didn't become better. Why? So maybe you can say that, oh, because genes provide a spectrum, and in that spectrum maybe you cannot go to more or less. But again, it's very important to discuss this in terms of this case. You cannot really generalize. This is only for these animals, for this group, and for this kind of test, and maybe you can get some different kind of results in another setting, in another species, including humans. So we cannot really generalize and make big conclusions from that. But it gives us a significant opinion that both gene and environment have a huge impact in our behavior, including intelligence or our risk of committing crime. And if we can understand these factors, probably we can help protecting these problems to occur. That is one side of the story from a social standpoint. From a biological or from a scientific standpoint, that is, I think, a good example for you to understand the effect of gene-environment interactions and how biopsychology is approaching in understanding behavior. I think that's enough for today, and we will, yes, questions, yes.
Do maize dull rats understand that there are maize brighter rats, or do they?
That's a good question. I don't think they understand. So I don't think they are aware. I think it's a question if animals have awareness or consciousness that was depending on the animals. For example, one case, depending on the experiments, chimpanzees have consciousness, at least conscious of self. They look at the mirror, after training at least, they can perceive that because they put a red mark on their face and they mirror in front of the animal, and then they try to touch their own head, and that means they are aware of themselves. But I don't think that that is the case for the rats.
Yeah.
Yes.
What happened with the dog with this impoverished cage? So you said that in enriched cage, the dull rats actually reach the level of bright rats, but what about impoverished cage?
There was no difference. So for the maize bright rats, they were good whether they are grown in the impoverished cage or enriched cage. They were both good. However, for the maize dull rats, environment was supportive if it is enriched.
On the previous slide with the graph, there was like a huge growth, I believe, in the maize dull rat.
This one?
The previous one.
This
yes.
one. This is the slide, yes.
Dull mice in the impoverished environments, so why is the force function so big?
Yeah, this is yes. Actually, I didn't see that the slide is here. You saw that. Yeah, this is exactly the slide. Yes. Let's discuss. So let's first come to the question of Andre. So this is the standard cage. This is the enriched cage. This is the maize bright rat in the enriched cage, and this is the maize this is the maize bright rat in the standard cage, impoverished cage. And this is the maize bright in the enriched environment. And the effect is actually more or less the same. It's not significantly different. However, the dull rats in the impoverished environment, they do a lot of mistakes. Sorry?
I think the right graph represents enriched environment and the left one standard.
No, this is the number of errors. So the more it is, the bad. This graph is sometimes, if you are not very careful, even I can mistake after so many years. I can be mistaken after so many years. This does not represent the success rate. This represents the number of errors. So the more you have, higher, that means the more errors the animal does. So here, the dull rats, do more errors when they are in the impoverished environment. However, when they are in the enriched, they are doing less errors. I would recommend you especially to compare them, then you can really understand. When you compare, the difference is huge, but when in enriched environment, the difference is decreased. But still they are not as good as, but they are still, you can consider as a significant improvement, and this difference is not so much.
So, and the scientists were still like feeding them, right?
What?
Feeding. They still get their food, right? So the dull mice, they just became very lazy because they knew they don't have to try to do anything because they will be fed anyway, right?
Food is about the interior of the cage.
Yeah.
How much toys are there?
I know more.
Well, food is another experiment.
So it is like this, they are grown in different environments, but whether the experiment is conducted in the way you ask or not, it is conducted similarly for two cases, two groups of animals. That's why actually as long as you test the animals under the same conditions, that's a controlled experiment and it doesn't matter the details in that case. Here what we should focus on, that they are actually, the only difference between them is their genetics. That's why they have 4 groups. And then they raise them, the dull rats are raised in the enriched environment, but the others also to test. Otherwise, you cannot compare. And based on that, yes?
I think the question was about motivation. And actually, when I read about these rat experiments, it says, well, there is food there, and the rat is trying to get the food. It doesn't mention how hungry the rat is.
Yes, but they are...
the rat is really hungry, does it perform better?
Yes, but they are you know, treated under very standard conditions, but of course maybe genetically some of them are more, you know, which is true because some of them may have a different genetics that could make them to have a different metabolism that could increase the way of their approach to food. But you know, you don't do it with one animal, you do a group of animals, and that situation is possible for all kind of animal groups. Other than that, of course, you do a controlled experiment, meaning that everything is very systematic. When you are going to feed them, it is systematically and same. What is the light day cycle? Everything is like very systematic and tight.
I understand. I mean, I just thought maybe do they have an experiment when they have the same conditions but different level of hunger? Does that influence performance? Because that's a question. Because if I think, think about rats, everybody thinks about studying at university, for example. I think, well, if you really want to get a good grade, or you just want it, but not so much, it probably influences your performance. Or how,
yes, of course. If you are not hungry, you wouldn't care. But probably they should have considered that. So I am sure they are not given food, so that they have to learn the food. But one thing I am sure, I think these differences shouldn't matter as long as the animals are kept under the same conditions. So maybe animals are hungry, maybe not. But as long as they assure that all of them are hungry or all of them are not hungry, then the experiment should be taken into consideration. However, experiments are always experiments, and the science is based on falsification. So we actually never believe the ultimate truth with a few experiments. We are always searching for a truth. So one important thing I would like you to also take as a lecture today is not to take science as a dogma. We just don't take one scientific finding and we don't rely on that for the rest of our lives. That's also important. Any other questions? Milana, you don't have any questions? No. Okay. Okay, if there are no questions, I would like to thank you for your attendance, and we will see each other again. Have a good day.